Hi, I'm Jamie Batts, your instructor for A&P 2. In the last section, we were talking about some of the, I want to say, like nitty-gritty reactions that occur in our metabolism. We're going to start looking at where the molecules for our metabolism come from. So let's go ahead and, I think it's this one. There we go. This next section is a review of digestion. Now, here's something that is super important to understand. There is a difference between metabolism and digestion, right? Digestion is actually breaking down the compounds. So these are hydrolysis reactions. This is where we're taking a large molecule and breaking them into smaller molecules, right? This can be done chemically through the use of enzymes. This can be done mechanically through the mixing and churning of the stomach and chewing, right, in our mouth. So either way, the end result of digestion is compounds that are small enough to be absorbed through our intestinal lining and go into our bloodstream. And once in the bloodstream, our cells can kind of pick and choose what they absorb based on concentration gradients and what they need to restore their nutrient pull. So we're going to review all of the steps of digestion. In the oral cavity, right, our mouth, our saliva is dissolving some of our organic molecules, right? Remember, salivary amylase, lingual lipase. There's also mechanical processing and that chemical digestion that takes place. In the stomach, we have a continuation of this mixing and churning, as well as protein digestion, beginning there with the use of pepsin. In the duodenum, we're introducing buffers to protect the lining of our small intestine. There are also enzymes from the pancreas as well as bile being injected into the chyme at this point. There are the several digestive enzymes that are there to finish the digestion of our molecules. In the jejunum and ileum, this is where absorption is occurring. It's not so much chemical breakdown anymore. The nutrients all go over to the liver through the hepatic portal system, and that's where they are either absorbed or converted. They're monitored, basically. So the large intestine's pulling all of the water out of our undigested food, as well as bacteria working in there to generate any other nutrients and vitamins, and everything is prepared for excretion. So this is an overview of the steps of digestion, a nice little image of where everything is occurring. That should be review for you. You can pause the video here, go back and review this section. We're going to get into carbohydrate digestion more specifically now. So, in normal resting state, carbohydrates are what our cells prefer to use for catabolism and ATP production. So if you're sitting here just studying, right, listening to me, not running around, or your heart rate's not elevated for whatever reason, your body's using carbohydrates for ATP. There are several steps to get the carbohydrates to the cells, right? So you eat a cracker, you eat a sandwich, you eat pizza, anything with carbs in it, pretty much anything. It's not like meat or vegetables. And you're going to begin this process. Salivary amylase is breaking down those very large starch molecules, complex carbohydrates, into disaccharides and trisaccharides. Disaccharides are two subunits, two monosaccharides large, and trisaccharides have three subunits to them. The amylase is deactivated and turned off once the chyme hits the stomach. For about 1 to 2 hours, it will be active, and then as that pH drops in the stomach with all the acid and secretions, it is deactivated and denatured. So in the duodenum and jejunum, there is pancreatic alpha amylase, which is secreted from the pancreas, and there's also brush border enzymes to complete the digestive process. These correspond with what we're learning in lab. So you have maltase, sucrase, and lactase. Maltase digests maltose, sucrase digests sucrose, and lactase digests lactose. And it tells you on this slide the subunits of those molecules as well. There's 2 glucose molecules in a maltose, there's a glucose and a fructose and a sucrose, and there's glucose and galactose and a lactose. So the glucose, fructose, and galactose are examples of monosaccharides, just meaning they're one subunit in size. The large intestine is going to take any of the cellulose, undigestible material, the fiber, right? It's going to use that to provide nutrients for the bacteria, and the bacteria can, in their process of breaking down the fiber, create intestinal gas, right? And this is just an overview of where everything occurs. We mentioned everything on here, CCKs, the release of all those things, secretin. Again, those are all just review hormones for you, GIP, right? So, how are carbohydrates absorbed and transported? Well, simple sugars go right through the small intestine membrane. They leave the cell through facilitated diffusion, and they diffuse right into the capillaries. So they're transported to the liver, through the hepatic portal vein. Very simple transport process here. They go into the blood and maintain normal blood glucose levels around 90, right? And the liver is going to take anything above 90. Once your liver is kind of like dipsticking your blood and saying, I think we got too much here, we're going to start storing the glucose in the form of glycogen, right? Glycogen is stored in the liver and skeletal muscles. So there you see the whole process in the lumen of our intestinal tract. That's the side here, which has the villi. There are those brush border enzymes breaking those disaccharides down into monosaccharides. And then they're simply absorbed through those intestinal cells, going through them and then into the blood, specifically the venous system, through the hepatic portal vein directly over to the liver, where anything extra is converted into glycogen. And the liver helps maintain our normal blood glucose levels. So how do we use carbs? Well, they're generally preferred, as I said, during resting states. Your body's using carbohydrates as the main form of ATP generation. Proteins and lipids are more important in cellular and structural components, so your body prefers not to use them. In skeletal muscle, you also have an excess glycogen store, and that makes sense, right? Glycogen's there, breaks down into glucose to give you energy in times of need. Your carbohydrate can be used and broken down during different times. So in most tissues, it's brought into the cells by a particular carrier protein. Glucose doesn't just zip right on through into your peripheral cells like water would. It needs a carrier protein there. And that carrier protein is regulated by a hormone called insulin. Once it's converted, or once it's absorbed, it can be converted into ribose or glycerol. If that glucose is needed for energy, then it's going to be converted into pyruvate, and the process of glycolysis will occur. Glycolysis produces 2 ATP molecules for every glucose molecule, and then pyruvate is used by the mitochondria in the citric acid cycle and electron transport chain to create 6 carbon dioxide, 12 water, and 34 ATP. That number of ATP has actually been updated since this book has been published. That's okay. You can memorize 34. That's fine. So this is just showing you how carbohydrates are used. Again, the skeletal muscle, resting, it's going to store the glucose as glycogen. When your cells need the glycogen, they can be converted back down into glucose, and glucose can go into the blood. As it's circulating in the blood, it will be absorbed by peripheral tissues. As long as insulin is present, insulin is activating the carrier protein, which allows glucose to come into the cell. If insulin isn't present, then glucose can't come into the cell. So guess what? Your blood glucose levels stay high. So guess what? You have insulin resistance, or maybe you don't have insulin at all. Your pancreas isn't producing it. You might have diabetes. So if that insulin is present and allows the glucose to come in, the glucose can come in and glycolysis will occur, which breaks our 6-carbon glucose molecule down into 2 3-carbon pyruvate molecules. You can stop the video here like I'm going to. This is a nice little intro to glycolysis. You can stop the video here, review those 3 questions on that last slide, and have an awesome day. Bye.